Hello friends, this is a part 3 video of system physiology animal notes. So last video we discussed with the blood and circulations, circulatory system and respiratory system. So this video I'll be explaining you the other important topic coming under this particular unit. So first we will study about the biosynthesis of neurotransmitter. So first acetylcholine, glutamate and your acetylcholine, glutamate, glycine, dopamine all will be originated from the presynaptic terminal, friends. So you should know from which particular part they are originating. Next, acetylcholine, the enzyme which catalyzes acetylcholine is choline acetyltransferase. The mechanism is that acetylcholine is synthesized from acetyl-CoA and choline. Next, glutamine, the enzyme involved in the synthesis is glutaminase, and here the glutamine amino acid will be converted into glutamate. Next, gamma aminobutyric acid, the enzyme which involved in synthesis of GABA is glutamic acid decarboxylase, and here the glutamate under the process called decarboxylation, it will be converted into gamma aminobutyric acid. Next, glycine, the enzyme which will involve in producing glycine neurotransmitter is serine hydroxymethyltransferase. So glycine will be produced from another amino acid called serine. Next is dopamine. Dopamine, the two important enzymes are being involved in synthesis dopamine. So tyrosine hydroxylase and dopa decarboxylase. So thyroxine hydroxylase enzyme will convert the thyroxine into dopa, and under the influence of this dopa decarboxylase, your dopa will be decarboxylated to form dopamine. Next thing is norepinephrine. The enzyme involved is a membrane bounded enzyme called dopamine beta hydroxylase. So norepinephrine will be produced from dopamine. Okay. So next is epinephrine. The enzyme involved is phenylethanolamine N-methyltransferase enzyme. So epinephrine is also synthesized from this norepinephrine, and the synthesis generally occur in the cytoplasm. Next is serotonin. Just like a dopamine, for the serotonin also two enzymes involved. First enzyme is tryptophan hydroxylase. So tryptophan will be converted into 5-hydroxytryptophan. Next enzyme is aromatic L-amino acid decarboxylase. So a 5-hydroxytryptophan will be converted into serotonin. Last enzyme is histamine. So the enzyme involved is histidine decarboxylase. So histidine will be decarboxylated to give histamine. So these are the biosynthesis of neurotransmitters. So here I have given you the mechanism, everything. So catecholamine includes a dopamine, non-epinephrine, epinephrine. So dopamine has been produced from tyrosine amino acid, non-epinephrine has been synthesized from dopamine, and epinephrine will be synthesized from non-epinephrine. So it is just like a coagulation cascade like that. Okay. Next is I had given, so with respect to glutamate, either it will be produced from alpha-ketoglutarate, that is Krebs cycle pathway, or else it will be produced directly synthesized from amino acid. Okay. The next thing is that neurotransmitter released by parasympathetic and sympathetic nervous system. Okay, so here I have given preganglionic neuron and postganglionic neuron. Preganglionic neuron will connect the CNS to the ganglia, whereas this postganglionic neuron will connect the ganglia to an effector organ. That's the difference. So sympathetic nervous system, you all know it activates the body to fight and flight response. Parasympathetic system means rest and digest division. Okay, so preganglionic neuron of sympathetic nervous system will secrete acetylcholine, whereas postganglionic neuron of the sympathetic nervous system will secrete norepinephrine, and the preganglionic neuron of parasympathetic nervous system will secrete acetylcholine, and the postganglionic neuron of the parasympathetic nervous system will be secreting acetylcholine. Okay. So next thing is that there was one question has been asked: which one of the following neurotransmitter is being secreted by preganglionic neuron of sympathetic nervous system? So preganglionic neuron of sympathetic nervous system will be secreting acetylcholine. So the correct option is acetylcholine. Next, the important topic is gas neurotransmitter. So gas neurotransmitter are classified into two important types: nitric acid and carbon dioxide, CO, that is carbon monoxide. Okay. Next thing is nitric oxide. So with respect to carbon monoxide, so first I'll tell you nitric acid. So nitric oxide is a paracrine signaling molecule, and this nitric oxide is synthesized from the amino acid called L-arginine, and the enzyme which is involved in producing nitric oxide is nitric oxide synthase. Okay, synthase, and many type of nerve cells will directly producing the nitric oxide in order to signal the neighboring cell. For example, nitric oxide that is released by the autonomic nerves in the penis will cause a local blood vessel dilation of the penis, which is responsible for the phenyl erection. Next, this nitric oxide is also produced by the activated macrophages and neutrophils, which is helpful for them in killing the invading microorganism. So this is the mechanism of nitric oxide production and synthesis and mechanism. Okay, so first the autonomic nerves in the blood vessel wall will release a neurotransmitter called acetylcholine. So this acetylcholine will act on the endothelial cells. So these endothelial cells will be lining the interior of the blood vessel, and this acetylcholine will act as a precursor molecule which will stimulate the synthesis of this nitric oxide. Then this synthesis may be triggered by the activation of another receptor, that is NMDA receptor. Okay, so NMDA means N-methyl D-aspartate receptor. Okay, actually this receptor is a type of G protein coupled inotropic glutamate receptor. So here GPCR, so GPCR is nothing but it's a NMDA receptor. So the synthesis of the nitric oxide is being stimulated or activated by this NMDA receptor. Okay, so this once this NMDA receptor has been stimulated, then it will be allowing the influx of the calcium ion. So this calcium ion will stimulate the synthesis of this nitric oxide. Okay, next so here you can able to see G protein coupled receptor. It will be so it will activate the phospholipase kinase. This is an enzyme which will cleave the phospholipid and produce diacylglycerol and IP3. So this IP3 will be acting as a secondary messenger, and it will be acting it will be increasing the amount of calcium ion. So here if you should know why calmodulin is present. So calmodulin is nothing but it's a calcium messenger binding protein. So it is actually a protein which will sense the increasing level of calcium, and it will be passing the signal. So once this calcium ion has been influxed or incorporated, it will activate the NO synthase enzyme which will produce the nitric oxide. So arginine will be converted into nitric oxide acetylcholine, whereas the produced nitric oxide will be entering into the effector muscle or effector organ. Here it is a smooth muscle, friends. So NO will be diffusing into the neighboring smooth muscle cell in the wall of blood vessel, where it will interact with another iron bound enzyme called guanyl cyclase. Okay, so this guanyl cyclase enzyme will be acting as a nitric oxide receptor, and it will produce the synthesis of cyclic GMP. So once the cyclic GMP will be activated, it will be leading to the process called vasodilation by activating the intermediate part called as PKG, that is phospholipokinase C. Okay, so this is what the thing, friends. So here you should know the guanyl cyclase catalyzes, that is this particular enzyme will catalyze the synthesis of cyclic GMP from GTP. So cyclic GMP is nothing but it's a secondary messenger. Okay, and it will modulate various effects in nervous system. Okay, so this cyclic GMP cannot able to act directly, therefore it will be activated by an effector part called as PKG. Okay, so this is what the thing that you should know, friends. So the increasing enzyme activity will be resulting in the synthesis of secondary messenger, which will include muscle cell relaxation and blood vessel dilation. So this is what the entire process. So please draw this particular thing in your preparation notebook. Next, the effect of Viagra. So first, this nitric oxide can increase the amount of cyclic GMP in the cytosol of endothelial cell as well as smooth muscle cell. But the thing is that the concentration of cyclic GMP will start to decrease because another enzyme called phosphodiesterase, which will cleave the cyclic GMP, and it will form an inactive form of GMP, that is guanosine monophosphate, that is 5-guanosine monophosphate. Okay, so sildenafil, which is a drug, this drug will cause the cyclic GMP level to remain high. How? It will inhibit the action of this particular enzyme. Ultimately, the cyclic GMP cannot be able to convert into an inactive form. So the level of concentration of CMP will be remain high in the smooth muscle cell after smooth muscle cell of penis after the nitric oxide production is induced by the local nerve terminals. Okay, that is autonomic nerves of the blood vessels. Okay, so this cyclic GMP causes blood vessels to get relaxed, and thereby penis will be erected. Next thing, a gas neurotransmitter which include carbon monoxide. Carbon monoxide is another type of diffusible gas that is endogenously formed in the central nervous system of neuron as well as enteric nervous system. So this carbon monoxide will be produced by an enzymatic degradation of heme by an enzyme called heme oxygenase 2, and like the nitric oxide, it is also acting as an intracellular messenger molecule, and it will be stimulating same soluble guanyl cyclase enzyme. Okay, and this CO can modulate the neurotransmitter, and it has been implicated in having role in olfaction, pain, long-term potentiation. Okay, and also it is endogenously formed, and it also essentially acts to attenuate the action of endotoxin released arginine vasopressin release, friends. So arginine vasopressin is nothing but it is a hormone that has been secreted by posterior pituitary gland of hypothalamus, which is involved in reabsorption of water from collecting ductal. Okay, so this carbon monoxide will stop the action of this endotoxin induced arginine vasopressin release. So the next type of neurotransmitter, they are classified in two types: excitatory neurotransmitter and inhibitory. First problem is that it will excite the neuron and cause it to fire off the messages, meaning that messages can be continuously passed from one cell to next cell, and the example include here glutamate, epinephrine, non-epinephrine. Next thing is here inhibitory neurotransmitter. As the name suggests, it will be either blocking or preventing the chemical message from being passed from one part of the cell to other part of the cell, or from one cell to another cell. Example include gamma aminobutyric acid, glycine and serotonin are the example of inhibitory neurotransmitter. Next, you should know the type of neurotransmitter. So amino acid, so include gamma aminobutyric acid and glutamic acid. Peptides, oxytocin and endorphin are the neurotransmitter belonging to peptide class. Epinephrine, non-epinephrine, histamine, dopamine, serotonin belong to the class of monoamine or catecholamine. Adenosine triphosphate belong to purine class, and nitric oxide and carbon monoxide will be coming under gasotransmitters. Okay, so next topic is blood-brain barrier. So it is a highly selective semi-permeable membrane lining the endothelial cell of nerves. Next, the small molecule will be passing through this blood-brain barrier by a mechanism called passive diffusion, whereas the nutrient, ion, organic anion or macromolecules like your glucose and amino acid will be passed through this blood-brain barrier by a process called active transport. Next, this blood-brain barrier will be permeable to water, carbon dioxide, oxygen, lipid, and also soluble substance like your alcohol, anesthetics, and also hydrophobic molecule like oxygen, carbon dioxide, hormone, and small non-polar molecule. It is permeable to all these kind of molecule, whereas this blood-brain barrier will be permeable to your plasma protein, non-lipid soluble organic molecule like large and hydrophilic molecule cannot able to pass through this BBB, whereas hydrophobic molecule can be easily transferred through this BBB. Next, transport of metabolic products like your glucose will be transferred through this, will be crossing this barrier using a specific transport protein or carrier protein, and this barrier also restricts the passage of peripheral immune factors like your signaling molecule, antibodies, immune cells cannot able to pass through the CNS. Next, the small molecule and their neuropeptide. So neuropeptide is nothing but they are also a type of neurotransmitter. They are auxiliary messenger molecule that always coexist in the nerve cell with one or more small molecule neurotransmitter. That is nothing but these are supplementary. Say if the neurotransmitter is not effective in bringing up the signal transmission, means then it will take the help from this neuropeptide. So always this neuropeptide mechanism is a co-release. It will be always released along with another neurotransmitter, and this neuropeptides, as the name suggests, they are vary in length but usually consist of between 3 to 36 amino acids. So here I had given you the neuropeptide. So what are the types or what are the compounds that has been considered neuropeptide? Include here substance B, cholecystokinin, enkephalin, somatostatin, neuropeptide Y, vasoactive intestinal peptide, neurotensin. Okay, and along with the neuropeptide are also given a neurotransmitter. So substance P is a neuropeptide for glutamate, GABA, acetylcholine, epinephrine, serotonin. All these neurotransmitter, if they cannot able to do their action correctly, they will be taking the help from the substance P. Whereas this GABA, dopamine, serotonin will be having this particular compound as their neuropeptide. Next, GABA, acetylcholine, dopamine, non-epinephrine, serotonin will be taking this enkephalin as their neuropeptide. Next, GABA, acetylcholine, non-epinephrine will be taking somatostatin as their neuropeptide. Next, non-epinephrine, serotonin will be taking neuropeptide Y, will be acting as a neuropeptide for these kind of neurotransmitter. Next, this vasoactive intestinal polypeptide will be helping the serotonin and acetylcholine, and the neurotensin is a neuropeptide which will be helping or supplementing the action of glycine neurotransmitter, acetylcholine, dopamine, non-epinephrine and epinephrine. Next is a glial cell and their function. So these are the types of glial cells. So first is astrocytes. This astrocytes helps in the regulation of external environment of neurons in the CNS, and they help to transfer the nutrient from the blood to neuron. Next is oligodendrocytes. These oligodendrocytes will form a myelin sheaths around the axons of CNS, whereas this Chapuan cells and neural LMO cells will form a myelin sheath around the peripheral axon. Next is microglial cell, and these are immune defense cell of the CNS, and the important process of function is that the phagocytes of foreign or degenerated material. Next is the ependymal cells. These will line the ventricles of brain, as well as these are present in the central canal of spinal cord, and the main function is that they are involved in homeostasis of CSF, there is cerebral spinal fluid, and they involve in brain metabolism, and also they are involved in clearance of waste from brain, and they are involved satellite cell and ganglionic cell. They generally support the neuron cell bodies with the ganglia of penis. Okay. So the next thing is with respect to phototransduction we are going to study. So before that, photoreceptor cells are classified into two types, rods as well as cones. So rods are used for the scopic vision or vision in dim light, and cones are used for photopic vision or vision in highlight. Next, the rods are very light sensitive, whereas cones are not light sensitive, and the loss of rods will lead to night blindness, but loss of cones will cause legal blindness. And these rods are not present in foeva, whereas cones are concentrated in foeva, and these rods are having slow response to light, whereas cones are having fast response to light, and these rods will confer achromatic vision, whereas cones will confer a colored vision. That is, achromatic vision means shades of black or grey. Next is the important process which every time at least one question will be asked, mechanism of phototransduction. So first I will be explaining you the mechanism undertaking in light, and then we will be explaining you in detail. So first understand this particular concept. So in the presence of light, the retinal, so retinal is nothing but this is what, friends. So rhodopsin is a G protein coupled receptor which is found on the rod cells of retina. So this rhodopsin is made up of two protein. One is retinal protein and another one is opsin protein. Both are involved in involved in getting the light and converting the light into an electrical signal. So this is the main mechanism. You should also know that this phototransduction generally occur in both rods as well as cone. So both the process are occurs in both as well as a similar process, friends. Okay, so the phototransduction is a process of converting the light system into the electrical systems. Okay, so it is made up of a protein called opsin and a molecule called retina, which is responsible for absorbing the light. Okay, so in the presence of light or in the light, the retinal is present in two important forms. One is all trans retinal, and it will be present as 11 cis retinal. So in the presence of light, it will be in a all trans retinal format. Okay, whereas in the dark, it will be in a 11 cis trans retinal format. Okay. So as I mentioned you earlier, so opsin will be one conformation, but after in a dark it will be in one conformation. But once the once the light has been observed, means it will be converted into a conformational change will be taken. This conformational change will activate another G protein coupled protein called transduction. This transduction is a heterotymeric G protein which is naturally expressed in rods and cones of retina. So once this transduction is been activated, it will activate a next enzyme called phosphodiesterase. As mentioned you earlier, this phosphodiesterase enzyme will convert the cyclic GMP into inactive form, that is 5' GMP. So this is a mechanism. So in the light, G protein, that is transduction will be in an active conformation. As a result, the phosphotransferase will also be in active conformation. As a result of this, if this enzyme is in active conformation, means so if this enzyme is being present, then ultimately it will be cleaving off all the cyclic GMP into inactive form. As a result, the intracellular concentration of cyclic GMP will be decreased in the presence of light. Okay, if the cyclic GMP concentration is started to decrease, then all the Na plus channel will be closed, friends. So if the Na plus channel is being closed, then no Na plus ions will be entering the cell. As a result, the positive ion concentration will be reduced and the cell membrane potential will be in a negative manner. So it will lead to a condition called hyperpolarization. So hyperpolarization is a change in the cell metabolic membrane potential that makes it more negative, whereas depolarization is opposite to hyperpolarization. It is the change in the cell membrane potential that makes it more positive. So under the stage of hyperpolarization, it will make the release of neurotransmitter to get reduced in nature, so that it will switching off the signal transduction mechanism. Whereas in the dark, the retina will be in the 11-6 conformation. As a result, in this conformation, it cannot able to activate the transduction. So transduction will be in an inactive stage and phosphodiesterase will become inactive. As a result, they cannot able to convert the cyclic GMP into 5' GMP. As a result, the intracellular concentration of cyclic GMP will start to increase in a peak. Once the cyclic GMP concentration will be increased, then it will open the Na plus channel. Then more number of Na plus ion will be entering into the rods and retinal cells. As a result, if more positive ion coming in, then the cell will be in a depolarization stage, where at this stage stage it will make the release of neurotransmitter to get high or increase. So let me explain you with the help of this particular diagram, friends. So here you can able to see what is given over here. So consider this as a retinal cell. So in the dark, the rhodopsin pigment will be there. So transduction will be in inactive stage, and the phosphodiesterase will also be in inactive stage. As a result, the cyclic GMP converting into 5-GMP cannot be occurred. So there is an increased number of intracellular GMP will be increased, and this dark also the Na plus channel will be open because the cyclic GMP acting as a secondary messenger, and it will open in a sodium ion, and more number of sodium ion will be entering into the cell. So here what is been happening, so the cell will be in a, so in the dark, this more number of potassium ion or sodium ion will enter into it. The cell will be in a depolarization stage, and also it will make the opening of voltage gated calcium ion channel, friends. So in the depolarization, that is depolarization of cell membrane will open up a voltage gated calcium channel, friends. As a result, an increased in intracellular concentration of calcium will be done. Okay, so this calcium will cause a vesicle which are containing the special chemical or nothing but neurotransmitter, and this vesicle will be merging into the cell membrane of rods. Okay, and it will be releasing the neurotransmitter into a synaptic cleft. So synaptic cleft is the area between the end of one cell to then another neuron, that is end of one neuron to another neuron. So I mentioned you here special chemical are being released. So the chemical name is an glutamate, since glutamate is a excitatory neurotransmitter. So that's the reason this increase of glutamate will be done in the process of phototransduction. So next thing is that under the influence of light, what it will be do means it will stimulate the rhodopsin, and it will activate the G protein called transduction. So here transduction will be in inactive stage, but once the light has been adopted by this rhodopsin, your 11-cis retinal will be converted into all trans retinal. As a result of all those process, the G protein will be in inactive stage will be converted into active stage, and it will be binding to the rhodopsin protein. So once it has binded, then this activated transduction will activate another enzyme called phosphodiesterase. As a result, the cyclic GMP concentration will start to decrease, and more number of 5' GMP will be increased, friends. So the inactive form will be started to increase. Okay. So, and also the opening of Na plus channel ion will be closed. So if this particular channel ion will be closed, means then the positive ions, that is Na plus ion cannot able to flow inside to the cell. It will make the cell to be in a hyperpolarization stage, that is cell membrane potential will be in a negative stage. So at this hyperpolarization stage, it will make the release of a neurotransmitter, that is here you can able to see at the light, the dark, it will be in a depolarization stage. The depolarization is a signal to increase the release of neurotransmitter glutamate, whereas in the light it will be in a hyperpolarization stage. So it is a signal to decrease the release of neurotransmitter, so that signal transduction will become in and it will be switch off. Okay, because it's a mechanism already in a light. Okay, so the next thing, another protein called arrestin will be there. So arrestin, it will be bind to the activated transduction, and it will be blocking the ability of rhodopsin to activate the transduction. As a result, the final cascade will be ending. So this is how arrestin will be make the transduction to become in a inactive stage. Okay, so this is a question I have been asked. When the rods of retina are kept in dark, were exposed to a light, phototransduction has been occurred. So following on some explanation given by the researcher regarding phototransduction. So they had given, first they kept the rods of retina in the dark condition, and then they had exposed to light condition. So what are the important thing that has to be occurred? So here you can able to see, please study this, at least this particular thing, so that it will be here. So first they kept the retinal in the dark, and they have then transferred the retina into light. So what are the mechanism that will be occur, like that they are asking. So here you can able to see, first thing is that activation of transduction. So when the retina kept in dark will be exposed to light, means ultimately the transduction will be active. So option number P, they are asking which one did not occur. So first I will explain you what are the things that will occur. So the transduction protein will be active. Next, inhibition of cyclic GMP phosphotransfer, no, there is no inhibition. It will be in an activated mode. So activated transduction will activate the phosphodiesterase. Next, closure of Na plus ion, yes, the Na plus channel will be closed once the retina kept in dark has exposed to light. Next, hyperpolarization of rods. Okay, so the correct option, that is in this particular question, which one of did not occur. So activation of transduction will not occur, like that they are asking. So which one did not occur? So So here I just mentioned you. So this question is little bit collapsing. So first you should write what are the thing occur. So generally when the rods kept in dark as exposed to light means activation or transduction will be done, closure of Na plus ions will be done, and hyperpolarization state will be experienced. Which one will not occur? They said that the inhibition of cyclic GMP phosphodiesterase. They say that the enzyme phosphodiesterase will be in an inactive mode or it will be inhibited. So this is the process which did not occur. So here at this stage the phosphodiesterase will be activated and more number of enzyme activity will be done. As a result, the cyclic GMP will be converted into 5-GMP. Okay, so this is the correct answer. So I hope this particular video will be very much helpful. If it is helpful, please try to subscribe to my YouTube channel. Thank you friends. Thanks for watching this video.